today's lab session we will focus on gene deletions, how we perform gene deletions using the Cobra toolbox. I will also show you this tool called Escher, which gives you a nice visual picture of what happens when you delete genes, how the rerouting of reactions happens and so on. And we will also look at how we perform single or double or synthetic lethal deletions, identify synthetic lethals using the Cobra toolbox. So, welcome back. Let us try out the following problems today in the lab. Let us identify single lethals in the E. coli core model using the single gene deletion function in Cobra toolbox. Let us identify double lethals in the E. coli core model using the double gene deletion, and a slightly more tricky problem is, can we find out a 4 to 5 gene deletion. So, remove 4 or 5 genes from E. coli without killing it, right. And I will show you there is an interesting website that can be used to visualize these as well, and try these for multiple models, not just the E. coli core model, but for the latest E. coli model or Mycobacterium tuberculosis and so on. These can be downloaded from the BIGG database, bigg.ucsd.edu. So, for deleting genes, it is good to check out the function delete model genes, right. And lastly, what would be the minimal non-essential reaction set in a given model? How would you compute that, right. So, a sort of minimal genome, right. So, if you remove any reaction, the organism is not going to survive. So, how would you come up with such a reaction set? So, these are some of the questions I want you to tackle today. So, this is a single gene deletion function. The outputs are growth ratio, growth rate on knockout. So, this is basically growth rate on knockout by growth rate of wild type, right, GR ratio, and what are the reactions that are released and deleted, and if there is any effect for the deletion, has effect. And you also have a single reaction deletion. Which is again very similar. You also have a double gene deletion, right. This essentially gives you a sort of matrix of, yes, the model has that information. So, if you look at these model.rxn gene mat, that is one place where this information is. I do not think it is there in rules. Yeah, rules also has this information. So, you have several reactions that have no rules, basically empty rules, and so this means that gene 31 and gene 32 or gene 20 and gene 21 are together necessary, right. Either these are necessary together or these are necessary together for the reaction to happen. So, the has effect is very useful. So, when you delete 32, it will actually check if it has an effect, right. At the gene level it may have an effect when you remove a gene, but maybe it affects no reaction, right. So, if you remove X5, it is not going to have any effect, assuming that it is not there individually in some other reaction elsewhere, right. If you remove X5, there will be no effect because X36 will be able to take care of that reaction. Has effect is one of the outputs. You look at help single gene deletion, the last output is has effect.
That is if it has any change, not just lethality.
No, if the reactions are going to change. So, look at the help. So, it says does a gene deletion affect anything, that is, are reactions removed from the model or not. And you can use either FBA or MoMA and so on.
There is only information on whether the gene is required for the reaction. There is no information on like how much it affects it. For example,
it is. So, the growth rate gives you the growth rate as well, and the general assumption is something is lethal if it is less than 5% of the wild type growth rate. So, if you look at, right, so it says it is 99.64% of wild type or 0.8 of wild type, 0.42, and yeah, for some there is no 0, For many you find it is 1.0, you find a few zeros as well. So this is the distribution. There are a few lethals, few sick deletions, few very lightly sick deletions, and many non-lethal deletions.
So will those in the middle also be considered as lethal?
So none of those are lethal. So this is the E. coli model. So let's just do the same thing on the E. coli model. It's going to take a little longer to do the single gene deletion because there are about 1260 genes in the E. coli model. You can download this model from the big database. You will see that double gene deletion is like much slower, the brute force. So you will have to use something like FastSL. And while this is going on, let me show you this nice web page. Actually, right here it's called Escher. So these are all, this is a visualization of E. coli, and you can see what happens when you start deleting reactions. You delete a reaction here, model map. There is a way to knock out the reactions. Let me check. You can have knockouts of reactions on Escher online. So you can just click a reaction to knock it out. So I knocked out some succinate dehydrogenase reaction or something. I've knocked out one more thing, and the growth rate is now 73%. Let's try to knock out something else. It's 3 knockouts, it's gone to 28%. 4 knockouts, it's gone to 26.5%. 5th knockout, it killed E. coli. So this is like a quintuple synthetic lethal set, right? So if you remove them separately, nothing happens. When you remove all 5, well, not necessarily. It could also mean that this is singly lethal. Yeah, but you know for sure that these are all not single lethals, right? So
You told that if it is less than 5% of what is maximum, then it is lethal. So the last one need not kill E. coli, right?
it must have. They must have done the similar check there. I don't know what's the exact cutoff they used. So this growth rate is 99.1. Yeah, so this might be a single lethal. So if you see, I can keep deleting these reactions, they're not going to make an effect. because these are not reactions that are currently being used, right. But now if I even delete one of these, it might already become lethal. Yeah, so this was a nice example. So I kept deleting multiple genes, nothing happened, but then finally when I deleted one, yeah, I think we have a synthetic lethal here. Delta PPC and MALS looks to be a synthetic lethal. Yeah, so this is a synthetic lethal. So PPC and MALS. So if you download FastAssay, you will also get these example matrices, I mean MATLAB files, which includes the stoichiometric matrix and the entire model actually.
So even for double gene deletion, you have the code, or you have FastAssay and you didn't integrate it with Cobra, or
It's not yet integrated with Cobra.
but the code is ready.
The code is here, the code is on GitHub.
Okay.
So you just have to download a separate package and install it. So how many single lethals in E. coli? How would you do it? Now, can you try out single reaction deletion? Is it much faster than what it is for me?
How a single reaction and single gene is making a difference?
In the sense?
Single reaction deletion, single gene deletion in the code. The code.
So reaction will remove reaction by reaction, gene will remove gene by gene and delete the corresponding reactions.
So
Some genes if removed may not lead to the removal of any reaction. If gene 1 is not connected to any other reaction, if gene 1 is just, if reaction 1 has a GPR which says like we saw here, wait, I think it's almost over. So, model.rules. Yeah. So this is 976 or 24. So if you remove 24, nothing will happen. Most likely.
It won't have it.
It won't have it. Okay, so this is an interesting point. So how does FBA work? It tries to maximize the growth rate through every reaction, right? So it doesn't actually try to account if there are like multiple copies of a gene, you can have a higher growth rate or any of those things, right? So you may, if that happens, see if you overexpress a gene, you can like basically double that value or something like that.
yes,
So
sir.
there's no way of changing. So if a enzyme is expressed by 2 genes, one gene expresses only a little amount. So if you remove that, technically it doesn't have an effect.
But if you remove the other one,
So all this is regulatory information, right? Which is not there in plain FBA. We will in future lecture we will look at efflux, which will essentially use this method. Efflux appropriately weights different, changes the LB and UB for reactions depending upon the expression levels of the genes. It helps you integrate say microarray data or any expression data set that you have with your flux model to make better predictions. So there are many methods to integrate, efflux is one of them. I am just running this again and let us see what Haas effect looks like. So, do you have an answer for the last question? What would be the minimal set of non-essential reactions in a given model?
Isn't it sparse optimal?
More or less, yes, you are on the right track even without attending today's morning's class.
So if you want to find how many deletions are not, like how many deletions will make a change, or can't we just count as effect the number of the sparsity of as effect? Will it not give whether a minimal reaction?
Good point, yes and no. The answer is no. Why?
So what is Haas effect? Does it give you lethality this thing, or
No.
just whether reaction is removed?
Whether a reaction is removed or not.
Then
Yeah. So you might be removing a non-essential reaction.
whatever output gives you the lethality coefficient. Is there an output there?
No, right. So that again, see your previous approach was a little better. Lavanya, you have an answer? You did this already, right?
We can non-FV and remove the fluxes remove the reactions which have maximum and minimum fluxes.
That is just removing blocked reactions, but beyond that, you can still remove some more reactions.
So even the single reaction deletion itself has some deleted reaction. It's returning deleted reactions, right? It is going to tell us which all reactions are deleted.
So that's just redundant. It will just give you R1, R2, R3, R4, Rn.
Is it the same as Haas effect, like putting Haas effect?
Yeah, for reaction there is no question of doing Haas effect. So does reaction have it? I was trying to remove it out of Cobra toolbox.
Right, it is.
It does have. What does the documentation say?
Haas effect. Does the reaction deletion affect anything? What does that mean?
It's just going to be all ones.
No, what if
Look at the value of Haas effect. I think it's just going to be all ones. I did try to edit it out of the toolbox. It's like we argued that it was redundant, and but we maybe kept it in for backward compatibility. So this is the code for single reaction deletion, right? And this is based on fast SL that we discussed a little earlier. So here the key idea is here. So if the reaction carries no flux in wild type, deleting it cannot affect the flux solution. See, not all reactions will carry a flux in the wild type solution, right? So corresponding to biomass optimization, given for any objective function, you run FBA and you get a flux solution, right? A flux distribution.
Because many technically only know what the solver is giving.
Exactly, right. So there are many equivalent flux distributions. The one thing that's guaranteed for you is that the function value is the same, right. So out of these equivalent flux distributions, which of them so can you use that to find out which reactions are essential and not essential. So there are some reactions which always stay 0, right, like your blocked reactions. So you can run a flux variability analysis, and if both the lower and upper bounds are, the maximum and minimum values are 0, you can discard those reactions. But beyond that, even out of a pair of reactions like we were seeing here, let me just reset this. you see that only, see MALS is not currently carrying a flux, right. So, but it could very well be carrying a flux. If I delete this, right, the growth rate has not changed for practical purposes, right, and this is carrying a flux instead, right. So, now if I go and touch this, it gets, you know, the knocked out and you have a synthetic lethal, right. But basically which of these do you pick initially? It is almost arbitrary, it depends upon the solver. Both these are equivalent states in which the cell can exist in reality, right. So, to come back, let us look at this part of the code now. So, if the reaction carries no flux in the wild type, right, in a FBA solution, in any given FBA solution, you can remove it without affecting the flux solution, and you can basically assign the wild type solution to the knockout solution without even computing or solving the LP, and that is where we say that has effect is false. There is no effect actually on deleting that reaction. So, if you run this, you will now see that this is much faster. Yeah, you can see this is way faster than the deletion that I was running previously, single reaction deletion. Right, so now oops. I think Haas reflection del Rxn are swapped. I thought I fixed this. Anyway, so there are 431 reactions that cannot be knocked out. So, if I start with a subset of this reaction set, so let us just
So, deleted reaction, the del reaction is the deleted reactions, right? So, 431 reactions can be deleted, right?
no, this was like a variable mismatch. This actually has effect.
Oh, okay.
The code has an error. Oh god, I have to put a pull request for this.
And sir, even then, it's like every reaction deletion will have a effect.
No, not all. That's exactly precisely what I was trying to tell you all along. Right? So some reactions when okay, let's just see. Okay, wait. So all these reactions don't have an effect. When you delete them, nothing happens.
Nothing happens in the sense, what are they trying to, like, I can't get the meaning of what is the meaning of nothing happens.
So that reaction is not carrying a flux. It is not currently important for the cell. So there are 2-3 possibilities. When I delete a reaction and nothing happens, there are 3 possibilities. What are the three possibilities? We discussed them before. At least two of them we clearly discussed them before.
It's not lethal.
No, the flux, the reaction is not carrying anything.
Yeah, but beyond that, that's like just saying, A equal to B equals A, right? So I want you to tell what is the reason that happens.
It blocked reaction.
It could be a blocked reaction. It could be a
What was that name?
It's not like a major name. It could be a conditionally blocked reaction, or it could be a gap or an unconditionally blocked reaction. So these are two possibilities. The third possibility is that it is not lethal, but that's also a possibility. It could be not lethal. I don't know if that's a mutually exclusive case. It's not lethal. The fourth possibility is that it could be part of a higher lethal set. When you remove a reaction out of a triple lethal set, nothing is going to happen. When you remove two reactions out of a triple lethal set, nothing is going to happen. Only when you remove all three, the organism is going to die.
So which one are they referring to here? Think they are referring to
Who's referring to?
like the as effect.
Has effect basically says removing the reaction changes, has an effect on the growth rate or not.
Also growth rate.
Okay, like single. It's like seeing whether that is singly lethal or not. That particular.
No, not lethal even. Whether it even
Changes growth rate.
changes growth rate or whether it even changes the reaction flux, whether it's even important, whether it is not unblocked. So it's actually a slightly debatable thing. Has effect for gene is the much easier thing to understand.
So if reactions A and B lead to reaction C and you remove A and okay, then has effect will be 1. It's like this.
The code is not wrong. I had given the arguments in the wrong order here. The gene deletion and reaction deletion orders are different. Let's just see. This is growth ratio, growth rate KO, growth rate wild type, has effect del reactions. This is growth ratio, growth rate KO, growth rate wild type, has effect del reaction. It's similar, maybe. Okay. Maybe the version I had was slightly different. This is del reactions has effect. That's where the problem is. Okay, so where are we? How much of this is done?
Single gene deletion is.
Have to try all of this. This is easy. Seriously easy stuff. You have to learn how to use delete model genes. What would be the naive way to do this? Find the minimal non-essential reaction set. The naive way to do is would be to remove a reaction. If it does not kill the organism, keep iterate. Then remove another reaction. If it kills the organism, revert that deletion, repeat. So in today's lab, we had an overview of how we perform gene deletions using the Cobra toolbox. I also showed you this interesting tool called Escher, which gives you a visual picture of what happens when you delete genes, and you can also see when the organism stops growing and so on. And we also looked at single and double gene deletions. In the next video, we will see how to integrate regulatory information into flux balance models and get better predictions. We will look at two techniques, one called regulated flux balance analysis or RFBA, and the other called E-flux, which gives a nice way to constrain the reactions based on transcription data or transcriptomic data.